So I've been pressed into service here as the moderator. And we have Diego is here to talk about Tock Trees, reconstructing the evolution dynamics of human tissues using fluctuating methylating clocks.
Okay, well, while they are setting up the slides, my name is Diego Mallo. I'm a research scientist working with Carlo here at ASU. And today I'm going to present a new phylogenetic method that is going to allow us to estimate the evolutionary dynamics of human tissues using methylation data. Yeah, there we go. So I don't need to convince this audience that cancer is an evolutionary process undergone by somatic cells. And that also somatic evolution is key to most pressing needs in cancer management. We need to be able to monitor, if possible, steer somatic evolution in what to improve patient treatment in what we have called evolutionary treatments that you all are experts and I'm not. We also need to understand the process of neoplastic progression and the origins of resistance. And for this, we need to understand somatic evolution. And last but not least, we need to try to predict the outcome of this evolutionary process of somatic cells to be able to stratify patients, focusing clinical resources in the patients that actually need it, and also to reduce the overtreatment of those that they don't need, treatment. So the phylogenetic method I'm going to be presenting today can be used to advance in any of these points, but it is more tailored to the last point and is the one we are especially interested in. We work with Barrett's esophagus, and we are trying to develop what we consider, we're trying to develop evolutionary biomarkers that we hope them to be universal biomarkers. So classically trying to predict the progression of premalignant disease, people develop what we call classical biomarkers that associate a specific somatic mutation or a number of somatic mutations with the outcome, trying to basically estimate progression to cancer. But this way of developing biomarkers is basically trying to reduce the stochasticity of evolution, trying to find mutations that have such a high or such a strong selective coefficient, basically such a strong selection that it is possible to predict the outcome given an evolutionary trajectory. Alternatively, we are trying to develop biomarkers that measure evolution itself, which we hope it is more general. Also, we have these kind of biomarkers have not been developed for such a long time, so there are probably more gains to be had, right? So in order to do this, we need to measure evolution, but it is very difficult to observe evolution in a human body, right? So alternatively, we need mathematical models of some sort to be able to, from one or a number of time points in which we get data, like reconstruct the past, the unseen clinical past, to be able to study evolution. So this was the title of my talk, basically. And what I'm going to be doing now is to kind of take it apart to show you exactly what we're talking about. So we are trying to reconstruct the evolutionary dynamics of human tissues using fluctuating methylation clocks. So what kind of human tissues are we considering? Where we are specifically focusing on human tissues that are organized in small populations of stem cells that can be sampled as a whole. In particular, we are interested in Barrett's esophagus, that is a premalignant condition, but this can be applied also to several healthy tissues, like, for example, colon, small intestine, and stomach. Which kind of data are we using? We are trying to use methylation data. And the main reason for this is that when you are trying to study the evolution of something that evolves slow, it's difficult to have enough signal to actually reconstruct a lot of parameters, right? And a lot of, generally speaking, healthy tissues and premalignant conditions evolve slower than cancers. So the classical somatic markers like SNVs and copy number alterations may not have enough signal to reconstruct the past well. So this is one reason why methylation is a good alternative for us, because it goes faster. Also, if we, like most, as you are familiar with, most methylation sites are going to be associated with the phenotype and not with the evolutionary process, just because of they are going to be regulating gene expression. So if we were able to, with one assay, obtain this data for both, tracking the evolution and tracking the phenotype, we could research into phenotypic plasticity, kind of two birds with one stone. Daryl Shibara, collaborators of us, had this idea some years ago of trying to find CPG sites, so methylation sites that evolve or get methylated and demethylated at random in a specific tissue. The reason is why in that tissue specifically, those markers are not being regulated. They are not regulating gene expression. And like him and many others, some of them here, I was not involved in this. Some like a year ago, put out this nice paper together in which they showed a way of finding these what they call fluctuating CPG sites that actually evolve like they were hypothesizing and can be used as evolutionary characters, basically, to reconstruct evolution. Usually what the kind of data we get, how we get this data is using epic arrays. We get an intensity ratio of methylated versus demethylated DNA copies in a given sample for each of the loci we are working with. And alternatively, we are working on targeted sequencing alternative that is not working yet. But that would be good to transfer the assay to the clinic because it would be much cheaper. Anyway, so the last point of the last component of this talk is the evolutionary dynamics we are going to be able to estimate given the data and the kind of system we have. And for this is going to depend on the model we are using, right? So what we are using is reusing a model from this previous paper I was showing earlier by Callum, also a member of this team, in which he put together a model that is basically described by, so this is basically representing the stem cell niche, and it's going to be determined by the number of stem cells and for each of the methylation sites, the fact that they are or they aren't methylated. This is going to be also determined by a number of evolutionary parameters that are the number of stem cells, the rates of methylation, the methylation and replacement rate. So when you have the data and the model, you just need to add one more step, that is the original state that we are assuming here, that is for half of the sites, they are fully methylated and half of them fully demethylated. We don't need to identify which ones. And with this, we can reconstruct all these evolutionary parameters. So this was originally done for one sample. This is basically equivalent to a branch of a phylogeny. So what we are doing here is extending it to reconstructing a phylogenetic tree using multiple samples instead of just one, reusing the same machinery, let's say. I'm not going to get into details here. That's basically kind of the translation we need to do from a single sample to the phylogenetic implementation with multiple samples. What is important to realize is that when we are doing this, we are basically reconstructing or studying evolution at two levels at the same time. Along each of the trees of the phylogeny, we are reconstructing the within stem cell niche dynamics, and the structure of the tree is going to give us the evolution of between the stem cell niches. So we will have, like crypts or glands that are dividing as they evolve. And at this point, we are using a model that is assuming that they are basically cloned, like duplicated, and we plan to relax this to be able to test different alternative hypotheses. So importantly, does this work? Well, what we did was to put together a simulation study with around 1,500 conditions. We tried to make it as realistic as possible, and I will show you some results. We can see here a simulated tree and an inferred tree. They look basically equivalent. Also, when we look at the data and the posterior predictive, they look very well, they align very well together. And for basically all of the estimate, for all of the evolutionary parameters, it usually works pretty well. There were a number of conditions for which this was not the case and they were not working well. But then we realized afterwards that actually was a problem that we had put together some simulation conditions that were not realistic because the demethylation rate and methylation rate were orders of magnitude different. And that's something we would never find in real data just because of these filtering steps we have, when we are generating or selecting these fluctuating CPT sites. So it's not a real problem for applied studies. And we also found out that elevated mutation rates may be a problem. And this is also something that is common with the single sample method and with any other phylogenetic method out there. So last but not least, I will show you some real data analysis to show you that the program works and the future directions. So we have analyzed results, real data from colon, small intestine, endometrium, and just one patient with Barrett's esophagus. So I will show you this kind of plots in which I have different patients in the x-axis and the estimated evolutionary parameter value in the y-axis. We will have usually 4 data points, 4 dots that indicate the reconstructed result using the single sample model, and a horizontal bar that is showing the result using our new method. So generally speaking, for this parameter, the results are pretty concordant. They make sense, and they are usually within the range. For the rates, I'm going to get into details here. We see the same pattern, but they are usually a little bit higher than the single sample implementation. What we think at this point is that it is because the phylogenetic implementation is leveraging the multiple samples and being able to soften a little bit the effects of saturation, but we are not sure. We are still doing some research on this. I will show you in the 40 seconds I have left some trees. We did reconstruct and see some kind of similar patterns between the different tissues. So in the case of colon and small intestine, we see, generally speaking, very long branches towards the tips. That is kind of expected because you could imagine that once the colon or the small intestine has grown into a full adult body, there is only replacement within each crypt, but crypts are not, like kind of taking over the whole colon, right? However, endometrium is different. There is more kind of evolution towards the present, which we could expect due to the, everything that happens with the menstrual cycle, and also with the Barrett's esophagus data. Just one case, we see a similar pattern in which branch lengths are much shorter towards the tips. So this is the comparison of what I just said. So just to conclude, I am presenting a new phylogenetic method to estimate evolutionary dynamics of human tissues organized in stem cell niches. It works well in simulation and real data. And we are going to be releasing it soon as a open source program. And the next step will be to be able to estimate changes in the evolutionary parameters along the phylogeny, basically. And that's all. I just want to thank funding sources, collaborators, and institutions, and the organizers for allowing me to be here. Thank you. And I'm happy to take any questions.
I very kind of cool approach. It makes sense, but how sensitive it is to the a priori assumptions about the stem cell niche? Because especially if you're looking at neoplastic issue, you're losing the structure of the niche, and there's kind of more promiscuity with sort of cells that can like easily differentiate. So how does this uncertainty impacts the inferences from your models?
Yeah, thank you. That's a great question. Actually, so potentially it would be quite sensitive, and the results may change in some parameters because there is a correlation between the number of stem cells and the stem cell replacement parameter that we are aware of. To answer your question more directly, in the future, we are planning to be able to estimate that S parameter, so the number of stem cells, along the phylogeny. So if it is just a case in which the number of stem cells changes, could potentially be detected, like using the estimation program. However, if it is a case in which the whole tissue kind of loses the structure and it's not organized in crypts anymore, then the program wouldn't work well, of course. Guess that's all.